students, this is our first lecture of Science and Technology in 10th standard. Let us start with our first chapter of Science 2. The name of the chapter is Heredity and Evolution. Students, in 9th standard, we have learned about heredity, mutations and the disorders occur due to the mutations. Can anybody tell me what is heredity? Yes, heredity is a transfer of biological characters, means the physical and mental characters from one generation to the next generation. And do you remember and can anybody tell me which component in the nucleus of the cell is responsible for heredity? Yes, it is the DNA. Let us revise some our previous knowledge and revise the structure of DNA and RNA first. First of all, we will see the DNA. Students, DNA is a double helical structure, means it is made up of two strands. Each strand of DNA is a polymer which is composed of monomer, and this monomer is a nucleotide. What is nucleotide then? The nucleotide is made up of a phosphate molecule, a sugar molecule and one nitrogenous base. The sugar molecule present in DNA is deoxyribose sugar. Here, nitrogenous bases are four types. They are adenine, guanine, cytosine and thymine. They are divided into two groups, purines and pyrimidines. Adenine and guanine, they are called purines. Cytosine and thymine, they are called pyrimidines. See here, students, the one strand of DNA has one nucleotide. They are linked together and forms a polymer. DNA has two strands, which these two strands are bonded together with hydrogen bonds. Adenine always pairs with thymine, see here, and cytosine always pairs with guanine. Adenine and thymine is paired with each other with double hydrogen bond, and cytosine and guanine, they are linked together with triple hydrogen bonds. Thus, the DNA looks like a ladder in which the sides of this ladder are formed phosphate and sugar molecule, and each step of this ladder is formed by a pair of nucleotide. It may be adenine and thymine pair or cytosine and guanine pair. Now see here the structure of RNA. RNA is also a polymer formed by nucleotide monomer. Here is the difference only, that is uracil nucleotide is present instead of thymine in DNA. RNA are of three types. mRNA, they are also called as messenger RNA, tRNA, transfer RNA and rRNA, ribosomal RNA. Students, this is the structure of DNA and RNA. You know that genes are present in DNA. Genes is a part of DNA. These genes participate in the functioning of the cell of the body, which takes the help of RNA. Students, all the information about protein synthesis is stored in this DNA, and proteins are synthesized by DNA, which are required for the body by the RNA. This DNA synthesizes protein with the help of RNA, and thus the proteins required for the body are synthesized in the cell of that living organism only. This process of protein synthesis takes place in cell and let us start with the process of protein synthesis. Let us learn with that the protein synthesis process. This process of protein synthesis is also called as central dogma. Means, what is central dogma? Central dogma is the process of producing mRNA, and the mRNA produces proteins which are required for the body, and they do all the functions of the body. This process is called as central dogma. Now, the process of protein synthesis is done in 3 steps. The first step is transcription. Students, the process of protein synthesis, in this process, the only one strand of DNA is used for this. The mRNA is produced with the help of one strand of DNA. The mRNA produces the strand or mRNA which is complementary to the sequence of the nucleotide on DNA. The DNA strand used in this protein synthesis has the complementary sequence of nucleotide with mRNA produced by that strand. This process of transcription is done first, and then next is the translation. In this step, the message of mRNA is read by tRNA. The mRNA produced in nucleus of the cell comes out of the nucleus. It comes in cytoplasm, and the message on mRNA is read by the tRNA. The message on mRNA is in the form of codons. Each codon has the proper sequence of nucleotides. Hence, it is called as triplet codon. Each triplet codon has a message for specific amino acid. Thus, the mRNA comes in cytoplasm. The message on mRNA or triplet codon is read by the tRNA, and a proper amino acid is supplied by tRNA. This process of reading the message and supplying the amino acid is called as translation. Now, the third step is translocation. rRNA, they are present in ribosomes. The amino acids supplied by tRNA, they come together, they bond together with peptide bonds and forms proteins. The ribosomes, which keeps on moving from one end of mRNA to the another end, and from one triplet codon to the next triplet codon. Thus, amino acid provided by tRNA at each triplet codon, they get bonded together with peptide bonds, and thus a protein is synthesized. This process is a translocation. Students, in this way, the proteins are synthesized in the cell. Students, we also learn about the mutations and various genetic disorders caused due to these mutations. Do you remember that? Can anybody tell me some disorders? Yes, it is sickle cell anemia, then albinism, then night blindness, Down syndrome, Turner syndrome, etc. Correct. Students, all living organisms produce new individual like themselves due to genes only. Some of these genes are transmitted to the next generation, and thus they brings the characteristics from parents to the offsprings without any change. But sometimes sudden changes occurs in these genes. These sudden changes are called as mutations. In these mutations, sometimes any nucleotide changes its position from one place to the another, and thus mutations occurs. Sometimes these mutations are minor, but sometimes these are considerable, and they causes various changes in offsprings. This process of mutation is everlasting, and one of the proof for Darwin's theory of natural selection and evolution. And we will see that theory of Darwin of natural selection in next lecture only. Thank you.